So our first speaker for today is Alexandra Tilivaki, who's a PhD candidate in the Schmitz lab at the Einstein Center for Neurosciences in Berlin, and the Parasubiculum lab, I hope that I'm pronouncing that okay, IMBB 4th in Greece. And in her most recent article, which I will post to the chat, she created an fast spiking basket cell model that incorporates nonlinear dendritic computations instead of just treating neurons as a one-stage integrator. And today she's going to be talking about something entirely different, but still related to neurons, which I'm super excited about because I am a total interneuron dork. So it's her favorite subject in addition to being mine. So please take it away, Alexandra.
Yeah, thank you very much, Emily. Hello, Neuromatch. Give me a second to share my screen and let's start. So, hello, I'm Alexandra, and today I would like to briefly present you a machine learning classification pipeline that can be used in order to identify the class in which a particular neuron belongs based on the electrophysiological values. The proposed approach can be used when, for example, immunohistochemistry information of the neurons is missing or very limited. So in our case in particular, we use this approach to identify the class for multiple neurons that are located in the parasubiculum. Very, very briefly, parasubiculum is a small elongated structure within the parahippocampal region lying between the presubiculum and the medial entorhinal cortex. Parasubiculum contains grid cells, head direction cells, and border cells. Okay, despite its promising role in brain function and its clear location, little is known about the cellular profile of the parasubicular neurons. Recently, Rosie Simons et al., a talented postdoctoral fellow colleague from our lab, Schmitz lab, identified the major neuronal classes of the parasubiculum and characterized their morphological and molecular properties. So specifically, parasubicular neurons can be mainly divided into three major classes, and these are the parvalbumin positive interneurons, the Reelin-positive interneurons, and the Reelin cells most of the times are inhibitory interneurons as well, and the WFS1 positive neurons that correspond to the parasubicular excitatory pyramidal neurons. So here you see some examples of typical electrophysiological values across the three main classes, the PB interneurons, the Reelin, and the excitatory WFS1 positive cells. The features were calculated after in vitro PaSClass recordings in iPad brain slices done by Rosie. And as we see, for multiple features, Reelin-positive and WFS1 positive neurons have very, very similar responses. So that's the first challenge of the dataset. So here is our challenging initial dataset. As we see, we have staining immunohistochemistry information for around only the half of the total around 570 neurons that Rosie recorded. From the rest neurons that we have also staining information. So only this data can be used for training, testing the performances of machine learning models. And from these, the vast majority, around the half, belong to the WFS1 positive neurons. The second presented class with staining information is the Reelin neurons, and the least presented class where we have staining information is the PV interneurons that are all 8 neurons out of the whole 500 neurons. So this is a typical multi-class because we have 3 classes data set, which is, as you see, very much imbalanced. We have training information for only the 50% of the data, and that means that we can understand what is the best classification algorithm that we can use to predict the remaining, shown in the green bar, only for the rest. And as I said before, we have one more challenge to face up, the fact that these two neuronal subcategories contain very similar approaches. So let's start to solve this problem. The first step that we do is that we use, through all the procedures, the Sklearn module in Python, which is a freely available open source module and with a great documentation, highly suggested people. We constructed and test the performance on several classification algorithms that are applicable for our task, which is a multi-class and imbalanced task. Very briefly, the classification models that we constructed and used were the Support Vector Classifier, the K-nearest network classifier, a Multilayer Perceptron Model, which is a typical artificial neuronal network, the Random Forest Classification, a typical multi-class version named complement of the Naive Bayes Classification, and the Logistic Regression. The first step for these models is to understand what are the best parameter values that can enable these models to perform well. And we tested this and we identified this by using hyperparameter tuning. Inside the Learn module is the grid-shared tuning. And we also used cross-validation throughout stratified K-fold. So we split our training and testing data sets into multiple tests. Where applicable, for example, in the random forest and in the support vector machine and in the logistic regression, we try to solve the imbalance issue by inserting class weights. So each particular class of the three, the excitatory and the two inhibitory population, we have a certain class weight for this to solve the imbalance. So as you see here, we train and test all the models. Here you can see, two representative performance metric score. The first is the very known, the accuracy score, and the second one is the F1 score, which is mostly used for multi-class and imbalanced datasets. And as we see here, in both of the cases, it seems like the Random Forest Classifier and the MLP, the Multilayer Perceptron Model, performs best. All the models we can say that have a good accuracy score, but we chose the two best, the RF for simplicity and the MLP. So you see here, first for the Random Forest Classification, the typical confusion matrix. The 0 corresponds to the WFS1 class, the 1 goes for the Reelin-positive neurons, and the 2 for the parvalbumin interneurons. And you see that our Random Forest Classification performs quite well. It has a good ROC curve. And impressively, the classifier does great jobs, especially for the parvalbumin interneurons, despite the fact that this is the least represented class. But it can also give us a good estimate and valuable predictions also for the two remaining classes, the WFS1 and the Reelin, despite the fact that these two neuronal subpopulations in the parasubiculum have very similar electrophysiological properties. We did the same also for the second best model, the multi-layer perceptron model, and you see here again the confusion matrix and the LOC curves. I have to admit here that this quiet graph is mainly used for binary classification, but we can do it for multi-class as well. And so we are pretty much confident here that these two models in combination can give us correct and successful predictions for the remaining population of our neurons where we do not have staining data. Here are also some more performance scores, precision, recall F1 in the AUC score, average, and I see this shown in the box plot across many random different trials. So we are pretty much confident that we can use these models. And here is the final version of our predictions. And I have to say here that in order to double check that all of our new predictions are totally correct, we run the two models, and the Random Forest Classification model and the Multilayer Perceptron, and out of 517 neurons, for the majority, more than 90% of it, our models have agreement in predictions. We have some mismatches for around 20 or something cells, and we excluded these cells for our data sets. So here is the final data set, and in summary, here is our proposition, not only for machine learning guys and computer scientists, but also for colleagues with a purely experimental and biological, molecular biological background like mine, that you can easily, don't be afraid, you can easily use machine learning when you don't have many staining data for your neurons. So we proposed this classification pipeline when the data are multi-class and imbalanced. For everyone interested, this whole approach will soon be available on GitHub. And I have to say that this work that I present you is a part of a bigger research project led by Rosie and supervised by Professor Dietmar Schmitz. And with this, I would like to thank you.
Thank you so much, Alexandra. That was a lovely talk. All right, so Pierre, if you have any questions, you can go on ahead. Otherwise, I have some questions.
Please go.
Oh yeah, so I was wondering, so I'm like roughly familiar with some of the machine learning like options. Why do you think that MLP and random forest seem to work the best for this particular classification problem?
I would say that MLP, by definition, it's a very powerful tool because it's an artificial neural network and it has a lot of parameters that can be tuned. For example, in the hyperparameter tuning that I showed earlier, we were able to select across different properties of this model. For example, the transfer function that we can use, namely a tanh or logistic or a ReLU function, or we can play with the number of hidden neurons and the number of hidden layers that this big neuronal network can have in order to solve our problem. So this means more computational capacity of this model. And for the random forest classification, it's one currently of the state-of-the-art classification models because it's one of the more robust classifiers that we currently have. Of course, we can even incorporate some other classification algorithms, for example, the LDA, the linear discriminant analysis. There are many out there that can be used. And I have to say that also the random forest we have also the flexibility to incorporate class weights. And because a major challenge of this data set was the imbalance, random forest, I think that could predict very well. But as we saw in the box plots earlier, all of the models have a quite high accuracy. So we don't say not to use the models, but we select these two.
Thank you. We have one question from the audience, and if you could answer really quickly, because we need to move on to our next speaker. Deletta asks, how many input data do you need from electrophysiology measurements in order to classify a neuron?
Oh, good question. So, for example, we have calculated around 20 or 21 specific electrophysiological values for each neuron, namely the half-width, the arrow base, the FH slope, the AHP, and so on and so forth. Usually, before entering a classifier, most of the people are first doing a kind of pre-processing on the data. And this kind of pre-processing can be, for example, a feature selection, which is to identify which of the values of the input data that we have a major significance. To our case, because we have a very imbalanced data set, we didn't do this. My answer is that this depends on the data set. If a data set is very clear and the particular neurons can be very well discriminated one type from the other, maybe you don't need many input data. Maybe you can do the same by using only 2 or 3 best electrophysiological features, but it depends. And I have to say, related to your previous question, one more thing for the random forest is that I consider this algorithm very well because it is kind of insensitive of data that are not so significant to data that are more significant.
Well, there's even more questions, but unfortunately we have to move on. Thank you again so much for that lovely talk.
Thank you.
All right, so our next speaker is Pierre Laurent.